This is a polypeptide chain. It is formed of multiple amino acids binded together to form a protein. A polypeptide chain will have an N-terminus at the beginning and a C-terminus at the end. These chains fold together in very complex ways to form functioning proteins. But before we get to that, let's talk about the structure of amino acids. Here is an amino acid that I've sketched out. The amino acid has an R group, which is what makes every amino acid unique, and its backbone, which is the rest of the sketch. The backbone has an amine group, a carboxyl group, and a single hydrogen over here. The amine group is composed of a nitrogen and two hydrogens. There could also be an extra hydrogen, but it'll be missing an electron. These amine groups bind to the carboxyl groups of other amino acids, and if there's no carboxyl groups to bind with, they will become the N-termini of the polypeptide chain. The carboxyl group is composed of a carbon and two oxygens, and a hydrogen. Sometimes the hydrogen can be missing its proton, in which case it would just be labeled as a single negative sign, because it would only be an electron. These carboxyl groups bind with the amine groups of other amino acids, and if they don't have an amine group to bind with, it'll become the C-terminus of the polypeptide chain. As you might have noticed, the variations of the amine groups and the carboxyl groups reflect each other. The amine group might have an extra proton, while the carboxyl group may be missing a proton. These two variations go together. As for the hydrogen up here, it is there to balance the amino acid, as this carbon in the middle needs to bind with 4 different things. Now, let's move on to the R group. There are 2 different types of R groups. There are hydrophobic ones and hydrophilic ones. The hydrophobic ones are very simple. They can be made of hexamer rings, or hydrocarbon chains. These 2 structures signify that the R group is hydrophobic. The hydrophobic R groups are repelled by water, which means they'll move away from it. The hydrophilic R groups are more diverse. First off, there's the regular hydrophilic ones. These R groups have a lot of nitrogen and oxygen in them. However, there can also be variations of the hydrophilic R group. These can have extra positive charges or negative charges. The ones with positive charges are acidic, and the other ones are basic. All of these are hydrophilic, which means they will be attracted to water. Primary structure looks like the structure that I showed you at the beginning of the video. It is the chain of amino acids that is called the polypeptide chain. It is held together by the covalent bonds formed between amino acids that are caused by the binding of the amine and carboxyl groups between the amino acids. These covalent bonds are caused by dehydration synthesis, which is a topic I covered in my previous video on macromolecules, so give that a watch if you don't know what that means. These covalent bonds are called peptide bonds because they're specific to proteins. Secondary structure occurs when the primary structure folds in on itself like this. When this happens, the folded structure is held together by hydrogen bonds that occur between the amine and carboxyl groups of different amino acids. These are not as strong as the covalent bonds that happen, but they're enough to keep the secondary structure together. Tertiary structure is when the secondary structure collapses in on itself because the secondary structure is surrounded by water. This water pushes all the hydrophobic R groups within the amino acids towards the inner side of the protein. And naturally, the hydrophilic R groups are pushed to the outside of the protein because they are attracted to the water. When this structure is folded completely, it is held together by hydrogen bonds and ionic bonds between the different R groups of the amino acids. However, these bonds can be broken by changes in temperature and pH. By this point in the folding, the protein is most likely functional. It won't need to go past this. However, some proteins can do quaternary folds. This is when multiple tertiary structures come together to form a protein complex. This is held together by hydrogen bonds and ionic bonds, just like the tertiary structure. However, these bonds happen between tertiary structures, not just within them. Not all tertiary structures go through this process, and very few need it to be functional. Tracing back to tertiary structures, some R groups can have sulfur in them. When two R groups that have sulfur come together, they can form a disulfide bridge. These bonds are much stronger and can resist most changes in temperature and pH. You'll find disulfide bridges in proteins that are exposed to very extreme conditions. That's all I wanted to cover in this video. Thank you for listening to my explanation. If you like how I explain these topics, please subscribe to the channel. I'll be uploading more videos like this soon. If you have any questions or are just confused, feel free to ask a question in the comments. I'll be sure to answer. And if you're curious about another topic, I'll be happy to answer that as well.